All right, now we're going to take a look at the process of glomerular filtration in a little more detail. We've already talked about how the glomerulus, which you can see here, this specialized ball of capillaries, um, is an extremely leaky capillary bed and is surrounded by the glomerular capsule, which collects everything that comes out, which is known as the filtrate. What we want to look at now is the balance of the pressures that allow this specialized capillary bed to be extremely leaky, but not so leaky that everything comes out and what's left behind in the blood vessels is too thick to continue to move through. So let's take a look at the diagram first to get oriented. Here we have a picture of the nephron with the glomerulus inside the glomerular capsule, the proximal convoluted tubule, the nephron loop, and the distal convoluted tubule. If we take a closer look just at this section, which includes the glomerulus, the afferent arteriole, and the efferent arteriole, the glomerular capsule, and then the proximal convoluted tubule that it leads into. First of all, I want to say that this region here is known as the renal corpuscle. So if you see that term, renal corpuscle, which really just means kidney body, corpus is body, corpuscle means little body. So that is referring to the glomerulus plus the glomerular capsule around it. And if you look over here to the left, where there's an actual microscope slide image of this, you can see why these were called the little kidney bodies. Scientists were looking at these long before they knew what they were. But in the slides, in the tissues, they could see this distinctive ball, and they could see the space around it. That's the glomerular space here. So this is the glomerulus, and this is the space between the glomerulus and the glomerular capsule. So in the renal corpuscle is where the balance of pressures that lead to glomerular filtration are so important. And there are two pressures that are key. Just like we saw with regular capillary exchange, the two pressures are hydrostatic pressure and osmotic pressure, specifically the osmotic pressure due to the concentration of proteins in the blood, which we call colloid oncotic pressure, or COP. Okay, so taking a look at this, the osmotic pressure, or the colloid oncotic pressure, the COP inside the vessel, is primarily due to the presence of proteins. And I mentioned in an earlier lecture that proteins are too large to leak out of the glomerulus. That means that the colloid oncotic pressure is helping to keep some water in the glomerular vessel. Otherwise, if all the water, if all the fluids leaked out, then we would just have this sticky mess of proteins and red blood cells and white blood cells. And it would become so thick that it wouldn't be able to make it through the glomerulus and back out the efferent arteriole. So the colloid oncotic pressure is important for keeping some water or some fluid inside the blood. However, it's opposed by hydrostatic pressure. The hydrostatic pressure inside the glomerulus is actually higher than what you would expect. And the reason is that the afferent arteriole, which you see here, is larger than the efferent arteriole, which means that it's letting more blood into the glomerulus than is allowed out. And if you imagine that, that this is a hose stretched out in a straight line, and the glomerulus is here in the middle, this leaky part with all the holes, if the blood is traveling this way, and I open up one side to let a lot in, but I close off the exit a little bit, it's going to tend to force substances out of the holes. Or encourage filtration. And that's exactly what happens. By making this a narrower vessel, it increases the hydrostatic pressure inside the glomerulus and helps to push fluids out, and is resisted only by the colloid oncotic pressure that keeps some of those fluids in. And in that way, the glomerulus is able to get everything out of the plasma and sent here to the proximal convoluted tubule for sorting. And we talked about in the previous lecture how we bring the good stuff back in. And we'll talk about how we bring the water back in the next lecture. But the point here in terms of glomerular filtration is to get as much out so that we can sort through it as possible. And this is done by arranging the balance between the colloid oncotic pressure and the hydrostatic pressure. Now, as we'll see in a moment, filtration, the rate of filtration can be controlled. When we need to speed it up or slow it down, we use the same idea that I just talked about with the afferent and the efferent arterioles being different diameters. If I need to increase filtration, I can increase the diameter of the afferent arteriole and send more fluid in, increasing hydrostatic pressure and increasing filtration. I would also constrict the efferent. If glomerular filtration is too high, then we can constrict the afferent, letting less blood in and reducing the hydrostatic pressure, and we can open up or dilate the efferent, and that will reduce the hydrostatic pressure and reduce glomerular filtration. Okay, so that's glomerular filtration in a nutshell. Now, we measure glomerular filtration by something called the glomerular filtration rate, or GFR. And if you've watched any of those hospital shows, or if you happen to work in a hospital and overhear medical professionals talking about a patient, they will say things like, oh, the GFR is too low. And that means that that usually means there's a problem with the patient going into shock and that filtration is shutting down. It's a sign that the balance of pressures in the body is not right. So how do we control glomerular filtration rate, and why is it important? So if we think about filtration producing a certain amount of fluid that's collected in a unit of time, in 1 minute. If we have a certain volume produced in 1 minute, that volume is passing into the proximal convoluted tubule. And the cells here, which you can see are lined with brush border so that they have lots of surface area to pull the good stuff back in, these cells can be overwhelmed by having too much filtrate pass through them. It's like having a conveyor belt in front of them that's going too fast and they can't pick out all the useful substances. So if GFR is too high, then we end up losing useful substances. They pass right through, we don't have a chance to bring them back into the body, and they go into the urine. Conversely, if GFR is too low, the cells of the proximal convoluted tubule pick out all the good stuff and reabsorb it into the body. And then they get bored and start picking up some of the toxic substances, the nitrogenous waste, and bringing those back into the body. So if GFR is too low, it results in a buildup of nitrogenous waste. If it's too high, then we end up losing stuff that we actually need to keep in our body. So GFR has to be tightly regulated. And the three mechanisms for regulation of GFR all use the same basic idea, which is dilating and constricting these two arterioles in order to change the hydrostatic pressure of the fluid in the glomerulus. Now, the first mechanism to control glomerular filtration rate is called autoregulation. And in this, it only responds to when the hydrostatic pressure of the blood, not so much in the glomerulus, but the hydrostatic pressure of the blood arriving at the afferent arteriole is too high. So the afferent arteriole gets this high pressure blood and it stretches. Well, anytime a muscle stretches, the smooth muscle in the vessel stretches, pardon my dog chasing the cat, the reflex is for the smooth muscle to then contract. So when high blood pressure arrives at the afferent arteriole and it stretches, it constricts in response, and that lets less blood through. And that prevents the hydrostatic pressure in the glomerulus from becoming too high and releasing too much fluid, also potentially damaging the delicate tissue of the glomerulus, which, because it's leaky, it's also very easy to rupture. So this is a protective mechanism to prevent any high blood pressure, systemic high blood pressure, from damaging the glomeruli. The next mechanism I want to talk about has a really horrible name. It's called the juxtaglomerular apparatus, and most people just abbreviate it JGA, but you do need to know the entire name. Juxta means next to, glomerular means glomerulus, and apparatus means thing. So it's the thing that's next to the glomerulus. Now, you may have noticed in all of the pictures that we've looked at of various nephrons that we have the glomerular capsule, the proximal convoluted tubule, the nephron loop, and the nephron loop always comes back and touches, joins where the afferent and efferent arteriole before it leads into the distal convoluted tubule. Same thing over here, proximal convoluted tubule, nephron loop comes right here between the arterioles and then leads into the distal convoluted tubule. That's because in this, which is the distal convoluted tubule that is coming back and nestling in between the arterioles, there are specialized cells. These cells are called the macula densa. And they sense the osmolarity, or the concentration of the fluid that's passing through this tubule here. And based on the concentration of the fluid, this means that the glomerular filtration is either just right, too fast, or too slow. The macula densa send signals to these little green cells that are actually wrapped around both arterioles. I know it doesn't look like that in this image. This arteriole we're looking at from the outside, so you can see the green cells all around it. This one the artist has drawn so that the vessel is cut through, so you really can just see a few of these cells. These are called juxtaglomerular cells, or JG cells. And they are extra smooth muscle cells that are on the outside of the vessel. So there's smooth muscle inside the vessels, in the vessel wall, just like the ordinary structure of an arteriole. But these JG cells are like an extra layer of smooth muscle. Now, if the macula densa senses that the glomerular filtration rate is too high, it sends a paracrine, or a local hormone signal, to the JG cells and tells them to constrict around the afferent arteriole and to relax around the efferent, which will reduce the hydrostatic pressure in the juxtaglomerular in the glomerulus. If glomerular filtration is too low, then the macula densa send a paracrine signal that causes the JG cells to relax. In other words, to help dilate the afferent arteriole, and will tell the JG cells to constrict or tighten around the efferent arteriole. And that will increase the hydrostatic pressure and increase the filtration. So we have the autoregulation, which is just a protective mechanism against high systemic pressure. But here we have a mechanism that allows for fine-tuning the glomerular filtration rate according to the fluid that's passing through. So it's like having a supervisor monitoring the process further down the line and saying, you know, okay, it's looking good, keep it the way it is, or it's too concentrated, we need to change the glomerular filtration rate, or it's too dilute, we need to change the glomerular filtration rate. So that's the juxtaglomerular apparatus. There is one other mechanism for controlling the glomerular filtration rate, and that is direct sympathetic stimulation. Here you can see a nerve that is connecting with the juxtaglomerular cells. This is a sympathetic nerve. When the sympathetic nervous system fires, it tells the juxtaglomerular cells to constrict, with the ones around the afferent arteriole in particular, and that's going to reduce the hydrostatic pressure and therefore reduce the filtration. So this makes a lot of sense because when your sympathetic nervous system is activated, it is when your body senses that you are in danger. If you are in danger, then your body is going to shift all of its resources to making sure that your heart is working optimally and that your skeletal muscles are receiving enough blood so that you can run away from danger. This is not the time that we want to be making urine. Your body is worried about getting you out of immediate danger, not worried about keeping your blood clean in the process. So in situations of high stress, where the sympathetic nervous system is active, urine production will actually go down because filtration is going down.